Hello everyone, you're welcome to this session where we are continuing with cell division. Lastly, we looked at the introduction to cell division. We saw different types of cell division, that is somatic and reproductive. And even we looked at cell cycle, whereby we said that if this is the cell, under somatic cell division, it is divided into G1 phase, which is the largest. We have the S phase, the G2 phase, and the M phase, which is the smallest. And we saw some cells in the G1 phase undergoing G0, cells like nerves. And we saw specifically that in the G1 phase, we see majorly synthesis of cellular organelles in the cytoplasm. Then in the synthetic phase, we saw majorly DNA replication, whereby if I have one DNA, it can replicate to form two daughter DNA. Then in the GAP2 phase is also a resting stage, where we saw in the GAP2 that more enzymes and structural proteins required for cell division are synthesized or manufactured within the GAP2 phase. Then lastly, we saw the M phase. And in this video, we are concentrating on the M phase of the cell cycle. And that is mitosis under somatic. And in short terms, to describe what mitosis is, that mitosis is the type of cell division whereby the original cell replicates or divide to form two identical daughter cells. We are saying they are identical. They are identical daughter cells having the same number. They have the same number of chromosomes as the original cell. That is what we can define mitosis. Type of cell division where the original or the parental cell divides to give two identical daughter cells having the same number of the chromosome as the parent. And these cells produced, they are known as diploid. They are diploid. Or they are diploid, not haploid. They are diploid. In somatic, we produce, or in mitosis, we produce diploid cells. And after forming diploid cells with the same number of chromosomes as the parent cell, we see that this mitosis has phases, whereby under mitosis, we are going to discuss phases whereby number one is karyokinesis. And number two, we shall find it is cytokinesis. Whereby under karyokinesis, we shall see it is divided into the mnemonic known as PMAT, whereby P stands for prophase, then metaphase, then anaphase, and telophase. So these are the four stages under karyokinesis, whereby karyokinesis, we are seeing the replication of the nucleus, or dividing of the nucleus into two, whereby this division of the nucleus takes place under prophase, metaphase, anaphase, and telophase. Then, after the nuclear division to form two cells, we see also the cytoplasmic division, whereby there is a creation of the cleavage furrow that divides to form two cells, and these two cells are formed during late anaphase and telophase, at the end of telophase. So that is where cytokinesis occurs. So without wasting time, we can now start to look at the different phases under karyokinesis. Then we end with cytokinesis. But I always remind you, before continuing, check if you haven't subscribed, please subscribe to this channel, share your comments, and always like the video when you enjoy. Always be reminded of that. So, under the stages of mitosis, we are saying we start with the prophase. If I have a cell, if we have a cell which has undergone interphase, that is G1, SC, and G2 phase, that is interphase. It has the nucleus with the nuclear envelope. It has the nuclear envelope. Then it is made up of also the nucleolus, and it has the chromatid. It has chromatin, and these chromatin are thread-like. They are thread-like and long, even thin. And we see at the stage where they have two asters, what we call the pericentriomas, or what you can call the asters. There are two of them. So this is after interphase. And it is the interphase that prepares for other stages like PMAT. Then from here, when the cell undergoes mitosis, and that is the first stage, is the prophase. When it undergoes prophase, in the early prophase, we are going to see that in early prophase, the nuclear envelope which was there during the interphase begins to disintegrate. We see then the nucleolus begins to disappear. We are seeing the nucleolus starts to disappear or to disintegrate also. Then we shall see the chromatin pattern that are thread-like and long. They begin to shorten and thicken to form chromosomes. So how many points we can note? In the early prophase, we are going to see the points to note is that the nuclear envelope disintegrates. The nucleolus begins to disappear, or the nucleolus disappears. Then we shall see the chromatin pattern condense and thicken. And then we see also the spindle fibers start to form. The spindle fibers, or the microtubules, start also to form at this point. They start to form, but they are really formed during the late. So when we go to the late prophase, that's where we are going to see now the envelope which had started to disintegrate. We see we are remaining with the fragments of the nuclear envelope. Then the spindle fibers which started to be formed during the early prophase, they are formed fully now. Then the chromosomes, or the chromatin pattern condense and thicken, and they have formed the chromosomes. So these are the chromosomes. And these chromosomes, if I can draw a structure of chromosome here, if I can draw a structure of chromosome, this chromosome is made up of the short arm joined together the center by the centromere. Then they have also the long arm. And these ones at them, we have the locus, different loci with the chromosomes, with genes, whereby the short arm is known as the P arm, then the long arm is known as the Q arm. And we are saying they are joined together the center by the centromere. This is the structure of the chromosome. And these chromosomes, they have a binding site where the spindle fibers attach to separate them. And this is called the kinetochore. So the kinetochore is a protein found on the centromere where the spindle fibers attach to separate the sister chromatids. So these two are what we call the sister chromatids. So the two sister chromatids join together at the centromere to form what we call the chromosome. So this is the structure of the chromosome which if you asked to draw the chromosome. So this chromosome, which is formed during late prophase, it is thick. It has become short to form the P arm and the Q arm, and they are attached by the spindle fiber. These are the what we call the spindle fibers, or you can call them the microtubules. So we are seeing them attached at the centromere by the protein known as the kinetochore. This is the kinetochore, and they are attached together or aligning along the spindle microtubules, but in the different positions. They are not aligning at the metaphase plate. So in the prophase, before we end, we are seeing the nuclear envelope at the end, it has disappeared. The nucleolus has disappeared. The chromatin, which were thread-like, have thickened to form the chromosome, joined together by the sister chromatids at the centromere. And we have seen also the spindle fiber formation. So don't forget that. And after seeing that, this is the guy that leads us into metaphase. And in metaphase, we are going to see under metaphase that we are seeing the migration of the asters to the opposite poles. But the only one note, the key feature to note is that at the middle of the cell, at the side, this is the pole, and this is the pole, and at the center, we call it the metaphase plate. Metaphase plate, or sometimes you can call it the equator of spindle. So in metaphase, the major point we are looking at is that we are going to see the chromosomes aligning. The chromosomes align at the metaphase plate. The metaphase plate. So that is the major point we can talk under metaphase. Metaphase is only that these chromosomes which are in different positions come and align themselves at the equator of spindle, or what we are calling the metaphase plate. So we see them aligning at the center, which we are calling the metaphase plate. That is what we can mention under metaphase. And the cells progress into anaphase, which is the third. And this anaphase, during early anaphase, in the early anaphase, we are going to see the separation, the chromosomes, or what we call the sister chromatids, separate, attached by the spindle fibers, attached by the spindle fibers. So we are saying this is the spindle fibers, these are the spindle fibers. So the spindle fibers, they pull apart to the opposite poles. So we are saying this is the pole and this is the pole. So the chromatids, or the microtubules attached at the centromere by a protein known as the kinetochore, they pull apart the chromosome and they separate the sister chromatids. And after separating them, that is what we are seeing in early anaphase. And by the late prophase, when we reach in the late anaphase, we are seeing these sister chromatids, they reach near the opposite poles. So in the early, they are separating at the equator spindle or at the metaphase plate. But in the late, they reach near the opposite poles. And remember, when they are separated, they form a V shape. They form a V shape being pulled towards the opposite poles by the microtubules, or what we are calling the spindle fibers. And when this is what we have seen, that under anaphase, after separation in late anaphase, the sister chromatids reach near the opposite poles, After reaching the opposite poles, the second major point is that the furrow formation, the cleavage furrow, is formed. So it is at this stage where cytokinosis begins. So cytokinesis is beginning here, whereby the cytoplasm or the cell is forming a cleavage furrow. This is the cleavage furrow that is being formed where the cells are going to separate. And it begins in the late anaphase. So in the late anaphase, the sister chromatids reach at the opposite poles and the cleavage furrow is formed. Those are the 2 points you have to note. And the late anaphase, then the cell progresses into what we call telophase, which is the number 4 stage and the last. Telophase, which is the last stage under karyokinesis, whereby under telophase, the cleavage furrow which is formed during late anaphase continues to constrict. Here it continues to constrict, and as it is constricting, we see the nucleolus which has disappeared during prophase begins to reappear. Then the chromatins which had condensed to form chromosomes also begin to lengthen. They begin to lengthen and become thread-like. Then the nuclear envelope which had disappeared during prophase begins to reappear. And as it reappears at the end of telophase, we are going to see cleavage at the cleavage furrow to form 2 cells. And these 2 cells, this is the first and this is the second, having chromosomes which are identical. They are homologous. They are identical and they are diploid. They are diploid, meaning they have the same number of chromosomes, having the same number of chromosomes as the original cell or the parent cell. So we are seeing that as we end, the nucleolus which had disappeared forms again. The nuclear envelope which had disintegrated now reforms, and then the chromatin pattern becomes again thread-like. It becomes thread-like. The nucleolus begins to also forms, forming 2 daughter cells which are identical to the original parent. This is what we call mitosis. So this is what we have discussed under karyokinesis, which is division of the nuclear material. So in summary, to remind you that this karyokinesis, which is division of the nuclear material, is composed of the PMAT, which is prophase. And this prophase, we have seen the major thing is that the nucleus disappears, nucleolus disappears, nuclear envelope disappears, spindle fibers are formed, and the chromatin pattern condense and shorten to form chromosomes, which are joined together by the 2 chromatids at the centromere. And we saw at the centromere, the protein which the spindle fiber attach is known as the kinetochore. Then we went into metaphase, whereby under metaphase we saw 1 point, major pregnant point, which is the aligning of the chromosomes at the metaphase plate, or what we call the equator of spindle. And then we saw in anaphase the sister chromatids being pulled by the spindle fibers. They pull apart and separate towards the opposite pole, and by the late anaphase, the sister chromatids form a V shape and are pulled and they reach near the opposite poles, and there is also formation of the furrow, and that's why we say the cytokinesis begins in late anaphase. Then we have also continued to see that in the telophase, there is the furrow continues to constrict, and then there is reappearance of the nuclear membrane, the nuclear envelope, the nucleolus reappears, then the chromosomes which had been formed, they lengthen to form again the thread-like, and the nucleolus reappears, producing 2 daughter cells which have the same number of chromosomes as the original parent. This is what we can talk under mitosis, under karyokinesis. Then lastly is, even we have talked about cytokinesis, which by cytokinesis has begun in the late anaphase by the furrow, by formation of the cleavage furrow. And we see in telophase being ended, whereby there is complete cleavage to form 2 daughter cells. So cytokinesis is the division of the cytoplasm to form 2 cells. And that one occurs concurrently with late anaphase and the telophase. So this one marks the end of mitosis. And what is the significance? You can always ask yourself, what is the significance of mitosis? This mitosis is very significant in our body, that whenever you get an injury, significance, whenever the cells get injury or they get damaged, we see being replaced. So replacement of dead, stroke damaged or injured cells is by mitosis, that in case you have wound, you have got an injury, what replaces the worn out cells is mitosis. And another major important is cell growth. All of us, we have grown from childhood to adulthood, and we see those somatic cells of the body, it has been by mitosis, whereby the cells proliferate or divide to increase in number, leading to growth. So these are the major significances: replacement of the dead or injured tissues, and also growth. And then lastly, we can look at the regulation, or what we can call control of mitosis, or what we call control of the cell cycle. How is the cell cycle controlled? This cell cycle is controlled majorly by two enzymes or proteins, whereby one is known as cyclin dependent protein kinases, what we abbreviate as CDKs. And then the second one is protein 53. And this protein 53 is a tumor suppressor antigen or marker, that is the one that regulates the cells when they are in G1. Is the one whenever we need more, it sends them to undergo other phases. And it is the same protein P53 that once no more growth is required, it makes cells to undergo G0 phase. And whenever there is damage in the DNA replication and the cell, this guy is able to repair, and in case repair fails, it can destine this cell for apoptosis. Apoptosis, that is programmed cell death. That whenever the cell, there is damage in the DNA during replication, there is some problem with during cell division, this P protein 53 can repair, but if repair fails, then it marks it for apoptosis, that is programmed cell death. So P53 is the one that prevents formation of oncogenes. So this guy is the one that prevents from formation of the oncogenes in the cells. Oncogenes, these are genes that causes uncontrollable proliferation of cells, and this one is the one that results into formation of cancers. So our body, when we are immunocompetent, we see we do not develop cancer due to presence of P53, which regulates that process. Hope you guys have learned something. Thank you for listening until the end. And I always remind you to subscribe, always share your comments, and like the video so that we can always continue to grow. Thank you so much.